Hi guys and welcome to my channel. Myself Dr. Akshay, and in today's session I'm going to be discussing about the topic nucleus. If you guys want to learn more about the cell, its structure and its functions, then click subscribe and the bell icon. So let's do this together. Coming to the topic nucleus. So the nucleus is the controlling center in the cell. So this nucleus sends messages to cell to grow, to mature, to replicate, and to die. This nucleus contains large quantities of DNA which comprises the genes. This genes determines the characteristics of intracellular enzymes as well as the structural proteins that helps in maintaining the cytoplasmic and nuclear activities. These genes also promote and control the reproduction of the cell. These genes first reproduce to create two identical sets of genes. Next, coming to nuclear membrane. This nuclear membrane is also known as nuclear envelope. So this nuclear membrane is a two bilayer membrane, and here one lies above the other. And here the outer nuclear membrane is continuous with the endoplasmic reticulum of the cell. You can see this is the outer nuclear membrane, and this outer nuclear membrane is continuous with this endoplasmic reticulum of the cell. Whereas the space between these two nuclear membrane is also continuous with the endoplasmic matrix in this endoplasmic reticulum. You can see this is the inner nuclear membrane and this is the outer nuclear membrane. The space between these two nuclear membrane is continuous with the matrix which is present in this endoplasmic reticulum. This nuclear membrane is penetrated by several thousand pores, and these pores are known as nuclear pores. So you can see in this figure, this nuclear membrane is penetrated by many pores, and these pores are known as nuclear pores. In these nuclear pores, large complex protein molecules are attached. You can see in this figure, protein molecule complexes are attached at the edge of this nuclear pore. So because of this attachment of protein molecule complex at the edge of this nuclear pore diameter is only about 9 nanometers. So this 9 nanometers of diameter is enough for the passing of 44,000 molecular weight molecules with reasonable ease. So next, coming to nucleoli and formation of ribosomes. So what do you mean by nucleoli? So these are nothing but one or more highly staining structures which are present in the nucleus. And this nucleoli does not have a limiting membrane which is seen in the nucleus. So this nucleoli is very free in the nuclear plasm. So the nucleoli is the accumulation of RNA and proteins which are similar to that which is present in the ribosomes in the cytoplasm. So this nucleoli becomes considerably enlarged when the cell is actively synthesizing proteins. So formation of nucleoli and of the ribosomes which are present in the cytoplasm begins in the nucleus. So firstly, the DNA genes which are present in the chromosomes will make the RNA to be synthesized. So some of this synthesized RNA is stored in this nucleoli, but most of this synthesized RNA are transported to the cytoplasm through nuclear pores. So this synthesized RNA which are transported to the cytoplasm will combine with the proteins which are present in the cytoplasm. So this RNA and protein combination will help to assemble mature ribosomes towards them. So after the assemble of these mature ribosomes towards them, this will help in synthesizing proteins which are helpful for the cellular metabolic functions. So that covers the topic nucleus. I hope you guys learned about this particular topic. Thank you so much guys. Love you all guys. See you guys in the next session.